Hello everyone, my name is Milan and today I will be presenting my project on how we can successfully express the green fluorescent protein in E. coli. These projects involves designing an expression vector, inserting the GFP gene, transforming bacteria and validating whether the protein is being made correctly. The aim is to use PET-42, a commonly used plasmid to drive high-level expression of a GFP and observe the results using fluorescence. Now project name and gene of interest GFP. Our goal is to express GFP in E. coli and confirm that it is being produced correctly. Green fluorescent protein is a protein that glows green under UV light. It was first discovered in jellyfish and it has since been used in research to track gene expression, study how proteins move inside cells, and create growing bacteria and animals for research purposes. The GFP gene is about 720 base pair long and it codes for beta barrel shaped protein that absorbs blue light and emits green fluorescence. This makes it easy to tell if the gene is being expressed simply by looking at the cells under UV light. Next, expression vector selection, PET-42. To express GFP in E. coli, we need a special tool called an expression vector, and this is a circular piece of DNA called plasmid that allows us to introduce foreign genes into bacteria. Why PET-42? The PET-42 vector is chosen for high-level bacterial expression of GFP in E. coli. I have chosen PET-42 because it is a T7 promoter which helps the bacteria produce lots of protein, and it contains an ampicillin resistance gene allowing us to select only the bacteria that have taken up the plasmid, and it has a C-terminal V5 and His6 tag, which makes it easier to detect and purify GFP later. This plasmid is commonly used in research because it produces high level of proteins and works well with the E. coli system. Now cloning strategy and primer design. Now that we have chosen our plasmid, we need to insert the GFP gene into it. This is done using a process called molecular cloning, which involves several steps. First, PCR amplification. We use a technique called polymerase chain reactions to make many copies of the GFP gene. Primer designs. We design special DNA sequences called primer, which helps copies the gene and also add restriction site to make it easier to insert into the plasmid. And the forward primer contains an NDL restriction site to ensure the gene is insert correctly, and the reverse primer contains a BAM-HI restriction site to help the gene fit into the plasmid properly. And ligation. After cutting the plasmid and gene with restriction enzymes, we use an enzyme called a DNA ligase to join them together. And final recombinant plasmid. The plasmid now contains the GFP gene and we are ready to introduce it into bacterial. Now transformation and selection. We have created our plasmid, so we need to introduce it into E. coli so the bacteria can start making GFP. This process is called transformation and we do it using techniques called heat shock. So how it works? First, mix the PET-42 GFP plasmid with component E. coli, which are specially prepared to take up DNA, and we briefly heat shock the cells at 42 degrees Celsius for 45 seconds, which makes tiny pores in their membrane and allows the plasmid to enter, and the cells are placed in a recovery medium so they can heal and start growing. Finally, we spread the cells on the LB agar plate with ampicillin. Since our plasmid has an ampicillin resistance gene, only bacteria that have taken up the plasmid will survive. To confirm which colony have successfully taken up the plasmid, we shine UV light on them. If they glow green, it means GFP is being expressed. Now verification of PET-42 GFP expression in E. coli. To be sure that the bacteria are truly making GFP, we perform three validation steps. First, plasmid mini prep extraction of recombinant plasmid. We extract the plasmid DNA from the bacteria and cut with the NDL and BAM-HI. We then run the DNA on a gel electrophoresis to confirm that the GFP gene is present. Sensor sequences. We send the plasmid for sequences using a T7 promoter primer to ensure the GFP gene is inserted correctly and has no mutations. And functional expression test. Fluorescent test. When exposed to UV light, bacteria producing GFP should grow green, western blot. We use antibodies to detect the GFP protein and confirm that it is being produced at the correct sites. These steps confirm that our experiment is successful. Now, last I am concluding. We have successfully designed an expression vector, insert the GFP gene in transform E. coli and validated GFP expression. Key takeaways. GFP was successfully cloned into PET-42 and insert into E. coli. Bacteria that took up and plasmid were selected using ampicillin resistance. Expression was confirmed using UV fluorescence and Western blot analysis. This experiment demonstrates the power of recombinant DNA technology, and GFP remains one of the most widely used tools in molecular biology, synthetic biology, and biomedical research. Thank you for listening to my presentation.